DNA recombination and gene transfer. So there's different roles of recombination. Two roles are DNA exchange and genetic diversity. Exchange of genes between two DNA molecules and contributes to population genetic diversity. You can generate new species in genetic engineering with recombination. There's three types of recombination: homologous, site-specific, and transposition. Homologous is nearly identical. It's between nearly identical strands, and you have site specific, which means you don't need a lot of similarity, and the specific sites allow for integration of a virus infection. Transposition is basically jumping element. Transpositions equal recombination. So it's when a DNA element moves from one site to another. You really hardly ever need any little sequence similarity, but the homologous you do need a lot of similarity. So there's two types of homologous recombination. You've got reciprocal general recombination, non-reciprocal general recombination. So in the reciprocal one, they'll give each other they'll exchange DNA, and in the second one, non-reciprocal, it's non-reciprocal because a host will give DNA to or the donor will give the DNA to a host in order to create a heteroduplex DNA. So recombination was won by Paul Berg right over here, and Gilbert and Sanger got a Nobel Prize for sequencing nucleic acids. Berg got the biochemistry of the nucleic acids and recombinant DNA right over here. Throw that right there, and we will highlight this. Oh, well, there you go. All right, reciprocal general recombination, exchange between two homologous chromosomes. So you have the nicking of the strands right here. Then the strands will exchange the DNA. The ligated strands will be joined together and move. The migration will cause a hybrid. You'll have a chi form, but what you have to do is you need to rotate this molecule. So you're just going to rotate it, basically like rubber bands, rotating it, and you're going to get a holiday junction. In the holiday junction, you will have horizontal or vertical cuts, which will eventually make the two recombinants. So essentially, the steps are nicking of strands, exchange of DNA, um, rotation of the chi molecule, ligating the strands together, and then creation of the holiday junction, and then endonuclease cutting these holiday junctions vertically or horizontally. This is a two DNA combined with a relaxed structure. So non-reciprocal general recombination, as you can see, it's one strand that gets incorporated into a chromosome, forming a stretch of heteroduplex DNA, which means like two different sources. This happens in bacterial transformation. When usually they take up naked DNA, the DNA is then integrated into the host right here. Site-specific recombination is virus. The phage DNA finds an attachment site. Very little region of similarity. You have the integrase, which puts them in, and then the virus will be put into the strands. Transposons, small segment of DNA which move from one region to another. They are inserted into different parts. So here's the thing: when you insert it within a gene, it will inactivate it because the gene will not express a protein. However, if the transposon is put in the chromosome, you'll be fine. You won't have any mutations. It is a mutation, but it's a silent mutation. Through evolution, you have transposons which carry enterotoxins and antibiotic resistance. When you have these transposons, you will have inverted repeats. So this goes G G G G T. This will go A C C C C C. This is not a direct complementation. So if it goes G G G G T, this should go starting with the T, A C C C C, which you can see right there. If you don't know how to do that, well, this goes that way, so you can figure it out. The transposons will insert themselves, and next to each other you will have inverted repeats. So there's three different ways to transfer genetic material: transformation, conjugation, transduction. Conjugation uses a pilicin's living cell-to-cell contact, and transformation is naked DNA of a living cell from a dead cell. You don't necessarily have to be dead. Transduction is bacteriophage changing, moving genes around. So the whole Griffith experiment was recombination. You had the S and the R cells. The R cells right here, they would be transformed into S cells because they would take up the S cell DNA. So transformation is naked DNA in a solution. Cells after die, they may give up their DNA, and then the living cells may take up the DNA. The cell receptor sites, competent cell receptor sites in the cell wall of the living cell may help take up the naked DNA, and so do competence factors. Transformation, naked DNA with cells. You have this naked DNA right here. It'll be broken up. Some will go in, the rest will be gone. Conjugation, direct cell-to-cell contact. These cells right here are being contacted by the F plasmid right here. The F minus lacks the pili, and the F plus has the sex pilus. So this is separate from the fimbriae. This is an F plus. This is for mating. The plasmid is sent through the F pilus. So the donor cells must be F plus, and plasmid is transferred through the pilus. Right here, F plus, sex pilus, plasmid is transformed. The F minus becomes an F plus. And the plasmid becomes from single-stranded DNA to double-stranded DNA. So DNA recombination, you have homologous exchange, which has reciprocal and non-reciprocal, site-specific, and transposons. And then the two ways we discussed today were transformation and conjugation.